ReadNet is a web tool for genomic research, which provides a convenient cloud control panel and an expression profile database of some brain disease and chemical exposure from microarray experiments. You can select samples from the database and generate a differential expression genes networks diagram, and then you might find out some information of relationships between the disease's chemical or pathway by comparing different gene networks. Here we provide a demonstration using the built-in database of ReadNet to generate the first differential expression genes networks diagram, which called the crowd. Step 1. Select samples. Open the source panel. Type in the keyword you want to search for microarray samples. Set the filter of gender, age range, or blank if you don't need any filter. Click the button Search to start searching, and then choose the samples for analysis. You can press Shift key to facilitate the selecting work in the list box. Click the button Add B to assign the selected samples to Group B, which usually referred as experimental group. Press the Kick button for driving out the old selection of list box. Select the samples and click the button Add A to assign into Group A, which usually referred as control group. The second step, samples grouping. If you haven't done the arrangement of groups in source panel, you could finish it or make the final confirm in the grouping panel. Here are the buttons for moving the selecting sample to the left side group, or the other one. You could sort the samples by age, tissues, or another variable from sample information. The combo box provides several algorithms of data pre-processing and normalization of biochips. You could change the selection and pay for a larger time-consuming grouping panel also provides the frequency distribution diagram and PCA plot of expression signal in both raw and normalized data. Users could ensure the quality control of samples by it. The third step, set the threshold for differential expression genes filter. To avoid a terrible giant network which might break down the whole view of webpage, ReadNet provides 3 common functions to perform differential expression gene filtering in graph panel, including student t-test, AUC value, and SVM scoring. After making sure that all the filters you want have been turned on, you could click on the scroll bar to adjust the threshold of each filter. The 2 scroll bars in the t-test meter box regulate different scales of threshold p-value. The lower scroll bars increase the p-value millionth per sliding, and the upper one increase hundredth per sliding. The scroll bar in the AUC meter box decides the threshold of the area range of ROC curve of differential expression gene. The final step, launch the gene crowd. After you finish all the setting of filter and samples, click the button Launch to post on the gene network. The gene network composed of many differential expression genes, which are generated by a single dataset, is called the crowd.